In today's episode, we're working through Chapter 5, Physical and Electrical Properties of Cells in the Nervous System. This is from our textbook, Neuroscience Fundamentals for Rehabilitation by Lori Lundy-Ekman. This chapter here is really foundational. Everything else you learn in neuroscience builds on it. So before we get into the pathways, the reflexes, movement, or sensation, we first have to understand what neurons and glial cells are doing at a physical and at a electrical level. When these processes work well, occupation is possible, but they really are fundamental. When they break down, we see weakness, sensory loss, fatigue, slowed processing, pain, and we also see impaired participation. So, as you listen today, I want you to keep one question in mind. What would this look like clinically if it stopped working? So, to frame our discussion today, because it's kind of a big one, let's briefly orient to the objectives for this chapter. By the end of this content, you really should be able to describe the four main components of a neuron and categorize sensory neurons, motor neurons, and interneurons using the structural classifications found in vertebrates. You should be able to explain the four types of membrane channels involved in neural transmission and describe how neurons maintain a negative resting membrane potential. You should also understand depolarization and hyperpolarization, compare local potentials and action potentials, and explain the two structural adaptations that can be used to increase conduction velocity. You should also be able to walk through the steps of an action potential, define afferent versus efferent, explain convergence and divergence, and identify the main types of glial cells in the central nervous system and in the peripheral nervous system. Finally, we're going to compare Guillain-Barré syndrome to multiple sclerosis and describe what neural stem cells are and what role they may play in recovery. With that roadmap here in mind, let's start at the cellular level. So this chapter focuses on three main types of cells. First are neurons. These are the nerve cells that are responsible for transmitting information. Second are glial cells, which support neurons structurally and metabolically. They regulate the neural environment and actively participate in both signaling and in defense. Third are the neural stem cells, which serve really as precursors to neurons and glia, and they play a role in learning, memory, and potential recovery. We'll begin here now with neurons. A typical neuron has four main components, and understanding these helps explain the direction of information flow. First are the dendrites. These are the branch like extensions that serve as the main input sites for the neuron. Most synaptic input from other neurons arrives at these dendrites. Next, we have the soma or the cell body. The soma contains the nucleus, and it's responsible for synthesizing a large quantity and variety of proteins, including the proteins that are used as neurotransmitters, which we will talk more about in the next chapter. From the soma extends the axon, which is the output unit of the neuron. Axons are specialized to transmit information away from the cell body to other neurons, muscle cells, or glands. Finally, at the end of the axon are the presynaptic terminals, which release neurotransmitter and communicate with the next cell or next neuron. So structurally, neurons are built for one-way flow, input through the dendrites, integration in the soma, and then output through the axon. Zooming in a bit, neurons are surrounded by a cell membrane, which separates the intracellular environment from the extracellular space. Some organelles are restricted here to the soma, including the nucleus, the Golgi apparatus, and the rough endoplasmic reticulum. And then other organelles such as the mitochondria, the lysosomes, and the smooth endoplasmic reticulum are distributed throughout the neuron, and this includes the axon. Neurons also rely heavily on cytoskeletal proteins. These specifically are microtubules, microfilaments, and neurofilaments. These structures maintain the neuron shape and they provide internal tracts for transport. And that transport is really critical for the function of the neuron, which brings us on to axoplasmic transport. Axoplasmic transport is the mechanism by which substances move along the axon. And this transport occurs in two directions, anterograde or retrograde. Anterograde transport moves materials from the soma toward the presynaptic terminal. This is essential for replacing proteins and organelles that are used or released at the synapse. Retrograde transport moves materials from the synapse back to the soma. Axoplasmic transport occurs at multiple speeds, and it slows with normal aging. It's also impaired in neurodegenerative diseases such as Alzheimer's disease, Huntington's disease, and amyotrophic lateral sclerosis. Now, this transport system matters clinically. A common chemotherapy drug is used to interfere with this microtubule dynamics in order to prevent cancer cells from dividing. However, the microtubules are also essential for axoplasmic transport. Because of this, 60 to 70% of individuals treated with this chemotherapy drug develop peripheral neuropathy. They then experience pain, numbness, and weakness that significantly affects their quality of life. This is a clear example of how disrupting cellular mechanisms leads directly then to functional impairment. So neurons can also be classified based on the number of processes extending from the soma. We have multipolar neurons, bipolar neurons, and pseudo-unipolar neurons. Multipolar neurons have multiple dendrites and a single axon. These are the most common neurons in the vertebrate nervous system. And a classic example is the spinal motor neuron that innervates skeletal muscle. Then we have bipolar neurons, which have one dendrite and one axon. An example of a bipolar neuron is the retinal bipolar cell in your eye. Next, we have pseudo-unipolar neurons, which have two axons and no true dendrites. These are typical of somatosensory neurons. Multipolar neurons are especially well suited for integration. A spinal motor neuron typically receives approximately 8,000 synapses on its dendrites and 2,000 synapses on its cell body. Okay, now that we understand a bit more about neuronal structure, we can move into how neurons actually transmit information. Neurons transmit information by undergoing rapid changes in electrical potential across their cell membranes. Electrical potential arises because ions, which are charged particles, they are unevenly distributed across the membrane. So electrical potential arises because ions are unevenly distributed across the membrane. When ions move across the membrane, electrical current is generated. To understand this, we need to speak a bit more about something called a gradient. So at rest, ions such as sodium, potassium, chloride, and calcium exist in different concentrations inside versus outside the neuron. Based on concentration alone, sodium, chloride, and calcium want to move into the neuron, and potassium wants to move out of the neuron. But concentration isn't the whole story here. The interior of the neuron is negatively charged relative to the outside. And because of this electrical gradient, sodium, potassium, and calcium are electrically attracted into the neuron, and chloride is electrically attracted out of the neuron. The combination of concentration gradient and electrical gradient is called the electrochemical gradient, and it determines which direction an ion wants to move. So even though ions want to move down their electrochemical gradients, the lipid membrane of the neuron prevents free movement. Ion channels then are specialized openings in the membrane that allow ions to pass through. Because ions are charged, their movement through these channels will then produce electrical current. Some channels are always open, while others are gated. Gated channels open in response to a stimulus, and they close when that stimulus is removed. And there are three types of gated channels. We have ligand-gated channels, voltage-gated channels, and modality-gated channels. So starting with ligand-gated channels, these channels open when a neurotransmitter binds to the channel. So the stimulus here is a neurotransmitter. Next, we have voltage-gated channels, and these open in response to changes in membrane voltage. So the change in voltage is the stimulus that acts on these voltage-gated channels. Next, we have modality-gated channels, which are specific to sensory neurons. And these open in response to mechanical forces, such as a stretch or a pressure, or in response to a temperature change. So in this case, with modality-gated channels, it is that mechanical force, the physical stretch or pressure or temperature change that actually acts as the stimulus, telling that channel to open. Now, in contrast to these gated channels, we also have leak channels, which are always open. And they allow a small number of ions to move across the membrane at a slow, continuous rate. Most leak channels are potassium channels. Leak channels play a major role in maintaining osmotic gradients and establishing the neuron's resting electrical state. So the term electrical potential refers to the difference in charge across the membrane, and it represents stored potential energy. That's why it's called a potential. It has potential energy. When ion channels open and ions move, that potential energy then becomes kinetic energy, moving energy. So neurons rely on three types of electrical potentials. We have the resting membrane potential, the local potentials, and the action potentials. Let's take these one at a time now. The resting membrane potential is the electrical potential across the membrane when a neuron is not transmitting information. It is a steady state condition with no net ion movement, even though individual ions do continue to move through the leak channels that are always open. The cell membrane functions as a capacitor that separates charge across the membrane. The typical resting membrane potential is approximately negative 70 millivolts, meaning the inside of the neuron at rest is negative relative to the outside. Remember, this is called a potential because it now has the potential to electrically change. Now, resting membrane potential is maintained by three main factors. First, we have passive diffusion of ions through leak channels, primarily potassium channels. Second, we have the sodium potassium pump, which uses ATP to move 3 sodium ions out of the neuron and 2 potassium ions in. Third, the negatively charged molecules that are trapped inside the neuron cannot diffuse through the membrane. As long as the neuron has ATP, an unequal ion distribution is maintained. When these ion channels open, the membrane potential changes, and that is where neuron signaling can begin. So when the membrane becomes less negative than resting potential, it's depolarized, increasing the likelihood that the neuron will generate a signal. When the membrane becomes more negative than resting potential, it is hyperpolarized, decreasing the excitability of that neuron. Now, small gradual changes in membrane potential are referred to as modulation. And these changes are the basis of local potentials. Local potentials include receptor potentials and synaptic potentials. Receptor potentials occur in sensory neurons when peripheral receptors are stretched, compressed, deformed, or exposed to thermal or chemical stimuli. These stimuli open modality-gated or ligand-gated channels, allowing sodium or calcium to enter the neuron. Synaptic potentials occur in motor neurons and interneurons when neurotransmitters bind to receptors on the postsynaptic membrane. Local potentials are graded, meaning that their size depends on the stimulus strength. They travel only 1 to 2 millimeters, decrease in amplitude with distance, and they can be strengthened through temporal summation or spatial summation. Because they travel such short distances, Local potentials alone really are not sufficient for long distance communication. And that is why neurons rely on action potentials. So, an action potential is a large depolarizing signal that is actively propagated along the axon. Action potentials are all or none. Once the threshold is reached, the action potential occurs with the same voltage and duration, regardless of the stimulus strength. So let's talk through the events of an action potential here. An action potential begins when summation of local potentials brings the membrane to threshold, which is approximately negative 55 millivolts. Remember, at rest, we're typically at negative 70 millivolts. So an action potential begins when summation of those local potentials brings the cell membrane to threshold, which is approximately negative 55 millivolts. This then triggers those voltage-gated sodium channels to open. Remember, voltage-gated means that the stimulus is that change in electrical potential. So the voltage-gated sodium channels now open, and the sodium floods into the neuron, and the membrane then rapidly depolarizes. The sodium channels then inactivate, and now voltage-gated potassium channels open. Potassium leaves the neuron, and the membrane then repolarizes. Because potassium channels close slowly, the membrane briefly becomes hyperpolarized. And we can talk about hyperpolarization in terms of something called the refractory periods. During the absolute refractory period, the sodium channels are inactivated and no new action potential can occur. During the relative refractory period, the membrane is hyperpolarized and a stronger than normal stimulus is required to generate another action potential. So during the absolute refractory period, that neuron is not going to fire because those sodium channels are not active. During the relative period, it would just take a much stronger than normal stimulus to get that neuron to fire because the neuron is more negative than that negative 70 millivolts. Now, once generated, the action potentials propagate by depolarizing adjacent segments of the membrane. The conduction velocity is increased by two structural adaptations of the neuron. One is an increased axon diameter, and two would be myelination. Myelin's a lipid protein sheath that insulates axons and it prevents loss of electrical current. In myelinated axons, the action potentials jump between the nodes of Ranvier, which lack myelin, and they contain high densities of sodium and potassium channels. So this node-to-node propagation is called saltatory conduction, and it greatly increases the speed of conduction. Okay, now functionally, neurons are classified by the direction they transmit information. First, we have afferent neurons with an A that carry sensory information toward the central nervous system. Then we have efferent neurons that carry motor commands from the central nervous system to the muscles and glands. In between, we have what are called interneurons, which process information locally, and they make up the largest class of neurons. We can also speak about communication and synapses, neuron communication in terms of convergence and divergence. Convergence occurs when multiple neurons synapse onto a single neuron, and divergence occurs when one neuron branches and synapses onto multiple neurons. Together, these processes allow information to be integrated and distributed throughout the nervous system. Moving on now from neurons to glial cells. Glial cells form a critical support network for neurons, and they also transmit information. And they're broadly categorized as myelinating glia. Glial cells, plural, can be called glia. Then there are the signaling, cleaning, and nourishing glia, and also the defending glia. So in the central nervous system, we have oligodendrocytes, and these myelinate portions of multiple axons. This is a myelinating glial cell. In the peripheral nervous system, we have Schwann cells, and they may fully myelinate one axon, or they could partially wrap several axons. They also provide trophic factors for axonal repair. So those were our myelinating glia, oligodendrocytes in the central nervous system, and Schwann cells in the peripheral nervous system. Next, we have astrocytes, and these regulate extracellular ions, neurotransmitters, nutrients, and waste. They also play a major role in forming the blood-brain barrier, which restricts the entry of substances from the bloodstream into the central nervous system. Next, we have microglia, satellite cells, and ependymal cells. Microglia function as immune cells. They help clear debris and they respond to injury. Satellite cells regulate the environment around neuronal somas. Remember, soma means cell body, and the peripheral nervous system. And they also can contribute to pain conditions. Ependymal cells line the ventricles and the spinal cord, and they help to produce and circulate cerebrospinal fluid. So those are our categories of glial cells. We had the myelinating glia, we have astrocytes, and then we have these microglia, satellite cells, and ependymal cells. So let's talk about what happens when some of the functions of these glial cells is disrupted. The primary issue that can occur surrounding these cells is demyelinating disorders. Damage to myelin, demyelination, disrupts conduction. Peripheral neuropathy involves damage to peripheral nerves and often affects the largest, most myelinated fibers. That can lead to pain, numbness, weakness, and impaired coordination. So that's just general damage to the peripheral nerves here. It can be the myelin in the peripheral nerves. Also in the peripheral nervous system, we have something called Guillain-Barré syndrome, which involves acute immune-mediated demyelination of peripheral nerves, often following something like an infection, with a rapid onset and usually a good recovery. So Guillain-Barré is caused by immune-mediated demyelination. This means that your body is attacking those cells that produce myelin. Remember, in the peripheral nervous system, these are the Schwann cells that are not functioning as they should be. This then affects myelin, leading to demyelination, meaning that things are not passed through neurons nearly as effectively as they should be. That leads to things like numbness, weakness, pain. We also have multiple sclerosis, one of the primary demyelinating conditions in the central nervous system. It's still immune-mediated demyelination, meaning our body is attacking itself. However, this time, because we're in the central nervous system, the cells being affected are oligodendrocytes. Then we see plaques in white matter and variable motor, sensory, visual, and cognitive systems. Now, a brief mention here of neural stem cells. Neural stem cells are immature, undifferentiated cells that are capable of self-renewal and, importantly, differentiation into new neurons and glial cells. In the mature brain, they contribute to learning and memory, and they represent an area of interest for rehabilitation and recovery. This is very much a new area of research, something we're still learning about and studying, but there's a lot of promise in the context of rehab and recovery when it comes to neural stem cells. Okay, so to summarize this chapter, we went over types of neurons, the directions that neurons can flow, or the direction of communication, I should say, of neurons, afferent versus efferent. We went over the different parts of a neuron. We spoke about the different types of channels on the neuron cell membrane. We went over different types of potentials, resting potential, electrical potential, local potential, action potential. We talked about different types of glial cells. And then we zoomed in a little bit on demyelinating disorders. All nervous system activity depends on the physical and electrical properties of cells. So understanding these mechanisms is what allows us as OTs to reason clinically to anticipate dysfunction and really at the end of the day affect these functions of our nervous system with a goal of achieving neuroplasticity to support rehabilitation and meaningful participation. Okay, thanks for listening. See you next time. Bye.